welcome to today's lecture. Thanks for coming. So today, as I said, we will look at neurons. It's a very interesting topic, I think. Some of the concepts that I will explain today are a bit complicated, but hopefully with all the schematics and stuff again, they will make sense. And I will tell you also some background stories about how they did some experiments on these axons that I will be talking about. So this is a famous model in electrophysiology. It's called the Hodgkin-Huxley model. These are two people. They won a Nobel Prize, I think, for this work. So the Hodgkin-Huxley model. So we'll talk about the nervous system today. So basically, if you have some electric potential, you have that this electric potential will now travel from the axon to your muscle, and it will tell it to do something, right? Some action. So the nervous system is a communication system formed by nerve cells or neurons. So we have that information is propagated along this long cylindrical segments that are called axons. And the way this is being done is basically through electrochemical signals. So this communication that is done in the nervous system happens between cells, and it occurs at junctions between what are known as synapses and dendrites. I'll make a drawing, probably will be bad, but let's see schematically. So basically you have here the cell body. Then you have these really long axons. This is the axon. And then basically this thing has a lot of junctions. It's like branches essentially. These are known as dendrites. And then basically at the end of the axon you have other things. Again, it's like branches, and then these things are called synapses. So basically you have that these signals get transmitted through these axons, and then they give some information in your body to do something, okay? All right, so this is now an important concept that I will introduce. This is excitability of neurons. This is what we will be doing today, basically. So what happens is that you apply some current to the system, and this can be small or large. We will be interested in the large current case. But if you just have a small current being applied for a short period of time, then the membrane potential will just return to its resting potential when you remove this current that you have applied. When the current is removed. However, the interesting case is if this current that you apply, again for a short period of time, exceeds a certain value, then you will get something that is known as the action potential. Okay, so we said that if you want to do something interesting, you basically, in this sodium and potassium pump that we have been talking about in the previous lectures, you need to expend energy, right? So this is this voltage or current that you're inputting into the system. Okay, so here I will write, but for a sufficiently high current, the membrane potential undergoes a large excursion So it will again return back to its resting potential, but first it will increase above a value and then it will become the resting potential. So this is called the action potential. Before returning to its resting value. Okay, so basically what you have is, if I were to draw this, and then we will derive that it looks like this, but for now I just want to give you an intuition for it. If this is the voltage you generate from the applied current, right? Then I'm gonna have this a function of time. Now I'm assuming that I don't have any spatial dependence, so I'm gonna have something like this. So what you do is you apply the current here, so you have some initial voltage, then the voltage increases, and then it will decrease to the equilibrium value. This is like the equilibrium. And we'll see why it does that later today. Otherwise, if you basically had a small current being applied, you wouldn't have this case of action potentials. What you would have, you apply against some current, which is smaller, and then it just goes to the equilibrium value instead. So basically what we will have, like after, when we do the more complicated things, that you have also spatial dependence, you will have that this kind of signal that you have with the action potential also gets transmitted like throughout your axon, and then that's how you get that spike that tells your muscles to do something, contract, for instance. Okay, so let me just say here that signals are transmitted by propagation of these action potentials. And for now, we will look at the case of a spatially independent model. So basically the way you do this kind of measurements is experimentally again, and you use a special type of mechanism experimentally. It's called basically the space clamp technique. So this can be measured or achieved in practice by inserting an electrode clamp along the axon to spread the current out. And this is known as the space clamp technique. It's actually super interesting when you read about this topic. Basically, Hodgkin and Huxley, the way they did these things were to use a giant squid. And the reason for that is that the axon of the giant squid is the largest one you can find in nature. Okay, so basically they removed the axon of this giant squid, poor squid, and then they took these electrodes and then they could measure basically these things, right? The current, voltage, and so on. So when I read this, I was like, oh wow, it's a giant squid. First of all, does anyone know how big giant squids are? They can grow up to 13 meters, 12 meters, and so on. So they're very big, right? So when I read, oh wow, it's a giant axon, it must be so huge. I was expecting a thing like this, I don't know. And then guess how much is the diameter of the giant axon of a squid? Just guess, says something random. Huh? Yeah, wow, you're good. It's like 0.5 millimeters. I was like, that's not so giant. I don't know, but then I was reading that other mammals have an axon that has a diameter of like 1 micrometer or something, so it's pretty giant. So yeah, but if you look at the length of this giant squid axon, is like it can grow up to like, I think 1 meter from what I read, but maybe I'm wrong. But in any case, the diameter is tiny still. Okay, that's the fun fact of the day. I don't have any other fun facts. All right, so let's get into the math. How do we actually do this? So I will make a schematic again of the cell membrane. So this is our cell membrane as usual. I will have an exterior side and an interior side. And in this example that I will do now, basically we have sodium ions on the exterior, okay? And I will be looking at this sodium-potassium pump. Yes?
It can be achieved in practice by something that you mean like
In experiments. Yeah, so like these guys that I was talking about, Hodgkin and Huxley, they just took this axon and put this electrode along the axon, and then they could measure these currents and stuff. So when I say in practice, I don't mean mathematically, like if you actually want to measure these things. Okay, so I'm talking about the sodium-potassium clamp again. So here I will have now potassium ions. And what we want to see is now you apply some external current. So I will call that I up, like applied current. And I will look at the outward current. So the one that goes from inside the cell outside. Okay, so that will be what we are solving for. Okay, so in this direction, from outside in the interior of the cell, I will have an external applied current. So this is I applied. And then I will be measuring, as I said, the outward current. So I will call it II. Okay, so let's say here what we did. So we are looking at the case where we apply a current, which is this external one from the exterior to the interior of the cell, to the axon. And what we want to do is, and we observe the ionic current outward that comes out. So I will call that, as I did here, I subscript I. So what we want to do is see how this responds to an applied current, and then say how much energy is being spent by this potassium-sodium pump. This is what we want to answer. Okay, so if you remember from yesterday, we had this equation. So I wrote C membrane, and then I wrote the rate of change of the voltage. And then this was basically I applied minus II. So we did this yesterday, right? When we were talking about capacitance and all these things. So if you don't recognize this form, the equation exactly that I wrote yesterday was basically C in dV in dt plus I equals to 0, right? So we had that I was minus the Q in dt, where Q was the charge difference. Does it look familiar? Okay. So I will call this equation 1. And now if I rewrite this, basically I can take the thing on the right-hand side on the left and write it as I minus I applied equals to 0. And this is the total outward current. Remember that II goes from the interior to the outside. This is the other way. Okay, and this thing here is the capacitance of the membrane. So the next step in deriving this Hodgkin-Huxley model is to basically see what are the equations that are being satisfied by the sodium ions and the potassium ions. So this is the next thing we need to do. But you already know how to do this from yesterday, so I'll just be using the stuff we set up in yesterday's lecture. So first I will focus on what happens to the potassium. So I will look at the potassium flow of ions. Potassium is a bit simpler than sodium. We will see why. So here, recall that we have a link between the voltage, so the potential V. This is the applied voltage. And the current, and the outward current. This is not the applied current, it's the one that you get as part of the solution, right? It's the emergent II. So here I will say that this is the resulting current. Okay, so from yesterday we had that the current for a specific ion species is given by Gs, and then we had V minus Vs, right? You remember this is the conductance, and then we said these are the potentials, basically. So we could relate them through that. And then we said that the gates that are open, basically for this conductivity, we can write it as Gs. This is given by n to the n, Gs max. This is again from yesterday. So this is the conductivity. And this was the number of open gates, right? So we could have also, sorry, the number of all gates. So you could have in a 2-channel case, 2 in your power here. If you have a 4-gate system, you would have 4 and so on. So we said that originally we saw it for the case of n Gs max, and we said you can generalize it to this. All right, so now that we have this, so n equals number of gates. We also saw yesterday that you can write down from the law of mass action an equation for how the number of open gates is changing with time, right? That was, if you remember, it involved the rates alpha and beta, and we rewrote it in the format of TV dn dt is equal to n. Right? This was the time scale at which you reach equilibrium. This is the rate at which the gates are becoming open, right? This is the equilibrium number of open gates, and it's a function of the voltage. And this is the number of open gates again. So this is equation number 2. And this equation, I should say here, is true regardless of the value of n. So whatever number of gates you have, it won't change this equation. So what has been found, again, experimentally, is that the potassium conductance may be controlled by this model when n is equal to 4. Again, this is a bit unsatisfactory, but essentially what they found is that you have, I will tell you why it's unsatisfactory. Basically, they found this. So this is for the potassium case. It looks like the system now for potassium consists of 4 gates, but it's actually an experimental fit so that they get sort of like the model to work. So this comes from an experimental fit. But the underlying physics, basically, they found that if you have this case of 4, it behaves like a 4-channel problem. So it's not really changing your physics. So they said it's a good number to use. So here, this n that I am using here will be called the potassium activation. So n is called the potassium activation. Okay, so this is the case for the potassium flow. You know how the potassium will be changing across this cell membrane. And we said, if I scroll now back to what we have written, you had this case. So we also need to know what happens to the sodium. The sodium is slightly more complicated in the sense that you have gates that are opening and closing and allowing the sodium in and out. So when you apply this current, basically you turn on or off your exchange of these sodium ions. So not only you have, as in this case, activation, you also have a deactivation mechanism. That's the difference. So we will have an extra equation for the sodium, and then we will form eventually for this model a closed system that can solve for all these things. The activation of potassium, sodium deactivation, and so on. So far, so good? Okay. All right, so let's do the same thing, but for the sodium flow now. So for the sodium conductance, there is a protein that turns the sodium current on. And another which turns it off. So if I want to compute the sodium conductance, this is going to be described by, now I will have Gs, the conductance of sodium, is given by G and A. This is what I have been calling Gs max. And I will have now, since I have both an activation mechanism and a deactivation one, I will have 2 things here. So I'm going to write 3 and 1. And again, these numbers, you shouldn't really care. Mathematically is what we need, but biologically, again, it's complicated. These things come from experimental measurements. So again, these powers are on experimental fitting, and they are not related to gates. So basically this M that I have been calling here is going to be the activation of sodium, and H is the deactivation. So we have to know, just like I did here, what did we do? N was the potassium activation, and I did an equation for the activation of potassium. We will have an equation that tells you how M is evolved in time and how H is evolved in time. That will tell you basically how the sodium is changing. Okay, so the two equations are exactly like what I have written there from the law of mass action. So we're gonna get tau mv, and then is the derivative of m with respect to time, the equilibrium minus m, and then exactly the same but for h. So I'm gonna have the time scale at which equilibrium is reached for the deactivation, and then it's gonna be h infinity v minus h. And this is equation 3 and 4 respectively. Okay, so this is an appropriate model. M here is called the sodium activation. And similarly, I have that, as I said before, this is the deactivation of sodium. So if we piece everything together that we know now, how the sodium is changing based on the conductance and how the potassium is changing, we can write down a full equation for the outward current when you apply an external current I applied, basically. So the next equation that I will write will tell you, from summing up of these different components, what is the total current outward, from inside the cell, outside. So this is the essence of the Hodgkin-Huxley model. For the outward ionic current. So I have, how did I denote that? It was I subscript I. And it's the sodium contribution. So I have, the sodium is this, so I am going to write m cubed h1. So remember from this equation, where was it? This one over here, I had the difference of voltages. So I'm gonna have here the voltage that we have minus the resting potential for sodium. So the resting potential for sodium, I'm gonna write as Na. And then I have exactly the same, but now I'm using the potassium one. The potassium one was this. So sorry, let me denote this by, no, this is not what I need. This is what I need here. So it's gonna be Gk, and then I have n to the 4. And then it's gonna be the voltage minus the resting potential of the potassium. And then this one, I'm not expecting you to know already, but basically what happens is that since we have the cell and we said we have many dissolved salts, the salts don't only include sodium and potassium, but they also have other ions, right, that are floating around. The main ions that you can find, if you look at these from experiments again, is chloride ions. And when you have all these applied currents and so on, there is some leakage that can occur. So these other ions can have some small contributions to the current that is going outward. So it's not gonna be the main component. The main component comes from sodium and potassium, but you also have some leakage. I'm gonna write now the conductance of that leakage is GL, and then it's just the potential minus the resting potential of all these other ions. And so let's write down what these three things are. This is going to be the sodium current. This is the potassium current. And finally, as I said, this third component over here is the leakage. And it's mainly Cl- ions. So this is chloride. So if you want to remember, just I will write here a small note. Other ions floating around contribute to the current. So let's name this equation over here, equation number 5. So when you want to see what are these components giving you as a function of time, you're going to have to solve a closed system of equations that is between 1 and 5. And equations 1 to 4 are ODs. So you can solve them. So this model comprises of equations 1 to 5, and then equations 1 to 4 are ODs. So basically, if I want to compute what is the resting potential from this system, the resting potential is the case when you have no current. So if I applied if there is no current in the system, you will have that this is 0. So this would mean that you have that the V is equal to the resting potential, right? So let's write this note down. So note, the resting potential is the value of V when the outward ionic current, which is II, is 0. Any questions? Okay. So now that we have set it up, we want to see how it actually behaves. And I'm going to basically show you that graph that I showed before, why it gives you that spike, and then you get the resting potential, okay? So there will be a lot of intuition that we have to think about when we are doing this now, so you will help me. So what does the potential actually do in this case? What's the interplay between the potential and all these activations of potassium and sodium? So to be able to see what happens in the model, I will have to tell you how to measure this equilibrium N, H, and M, so the M infinities and so on. And I will also have to tell you how the time scale at which you reach this equilibrium is reached. So this you don't know, I have to give it to you. So this is again measured experimentally in a lab. But we know exactly the form of these functions as a function of the difference between the potential and the equilibrium potential. So this is what I will draw now, so I need different colors. And I will put a disclaimer here. Basically, these graphs are obtained from experiments. So I'll make two graphs here. They will have the same horizontal axis. And one of them will be so this is graph 1. And it will be the equilibrium values. And then graph 2 is going to be the time scales at which you reach these equilibriums. Okay, so let's see what we have. So first I will draw the deactivation. So this curve looks like this, goes to 0. This is H infinity. And I know that the time scale at which this happens basically looks like this. I have something that grows and then it reaches some value. this is going to be tau H. So this is the deactivation of sodium. Now, I also have N. N is the activation, if you remember, of potassium. So I'm gonna draw this is a non-zero value initially, and it grows in some pattern. So it's gonna be N infinity here. And I know that the time scale at which this happens basically looks like this, but then the two approach the same value. This is tau N. If you can distinguish my H and N, let me know. Okay, and then finally I have M. M basically looks like this. I reach some value. This is gonna be M infinity. And then finally I have that the time scale for this one is very short in comparison to the other two. So it's gonna be tau M. Okay, so this first one, just so that you know, is going to be, we said, the equilibrium, so equilibrium gate variables. You should remember that is the equilibrium from the infinity sign here as functions of the potential relative to the equilibrium one, which is the resting potential. And then for this one, I'm not gonna repeat all of it, but it's basically the time scale, so the relaxation times. Again, as a function of the potential relative to the resting potential. Okay, so this, I wasn't expecting you to know, is going to be given to you, but now we can see how the potential is changing based on this. So there are 6 steps that are involved. And then using our equations, we will be able to see this action potential in practice, how it changes with time. So I have 6 steps. The first step that you should do is apply this external current. So that you do in the experiments. So you apply a current, and you also do it in the mathematical model. So you have some I applied. All right, so once you have done that, then you have that the voltage, so this is the applied voltage in the system, rises due to this applied current. How do I know this? So, well, you know that from equation 1. So probably it's covered, so I will rewrite it here. So equation 1 was this one. So it was I minus I applied. So if I write it in the opposite way, basically you have I applied minus II. So this is our equation 1. So this means that at the first time instance, for instance, you put this applied current, right? So this is going to be a positive value. So this means that this dV dt will increase as well, right? So this means dV dt is positive, is going to give you an increasing V. So that's why I know it rises. Now, step 3. So if you look at this graph that I drew here, you see that tau m, the relaxation time for the activation m, is much smaller than the other two, right? So what we have here is that tau m is small. This is graph 2. So what this means is m rises quickly. And you can see that clearly from equation 3. Again, I'm not expecting you to just remember it, but if you just look at your notes, if you rearrange equation 3 by dividing by the relaxation time, what you're going to see is that you have m infinity minus m divided by tau m, right? So what this says is that if tau m is very small, this thing is going to be large, right? So this is going to rise very quickly. This is what this equation is saying. So m was the activation of sodium, correct? So this tells us that the sodium ions if this rises very quickly, they're gonna flood into the axon from the outside. From the outside. Okay, so now what is the fourth step? The fourth step, again, I will now be looking at the same equation, so equation number 3, and from that we know that this causes even more of a potential difference. And this causes basically the M infinity to rise. So this you can see from graph 1 over there. So this is graph 1, which causes, is basically they are all like interconnected. So this also causes even more of a potential difference. And this I know exactly from the point above, from step 3. So essentially what I'm saying here is if you look at equation number 3, which is this one, I'm saying that you're causing M infinity to increase. So if this increases now and everything else is the same, this also increases, right? So again, it increases your M, and so it causes even more of a potential difference. This is what this is saying. Okay, great. So once I have that, then in step number 5, now if you look at the graph over there, what we are saying is that we are increasing M infinity. M infinity is the activation of sodium, right? So while you are increasing the activation of sodium, you're decreasing the deactivation, which is H. So in graph 1, again, the H is the blue curve, it goes down, right? So here, H infinity, the deactivation, is going down, so it falls, which means it causes the sodium ions, causing the sodium ions to flow slowly as the conductance falls. So here you have that the gates are essentially closing. So while this is happening, we also have that N infinity. So N infinity, there is the red, rises. This is again from graph 1. This causes, so N is now the potassium, so causes potassium ions, bless you, to flow out of the axon. So here what you have is basically the G of K now rises, which means that the gates open. Okay, so while this procedure is happening now, this causes an overshoot. Because you're causing these ions of potassium to flow out of the axon, you have an overshoot of potassium past the resting potential. And this is because the potential of the potassium is less than this. So this is the resting equilibrium for potassium ions. Okay, so once this has happened, then we also have, because now you overshoot above the equilibrium or resting potential for the potassium, you're gonna have a leakage of these extra potassium ions, right? So you have more than you need. So it's gonna be a slow leakage of potassium ions back into the axon. causing the system to go to equilibrium. Basically, this is like a recovering mechanism from the overshoot. This is all this step is saying. Okay, and then this basically concludes the whole process that is happening in the Hodgkin-Huxley model. So if I were to draw a diagram of this action potential, this is what you're seeing. So I'm going to draw in the horizontal axis here. This is denoting basically my resting equilibrium potential. And I'm going to have it as a function of time. And this axis is the applied voltage or applied potential, okay? So what you do, it was the step one. What was step one? To apply the external current, right? From the exterior to the inside of the cell membrane. So at time 0 here, I'm going to apply some current that will induce a voltage, right? This is what we said. So this is the initial value. Then I have, from the next step in our process, that you have sodium gates. So again, sorry, let me write it with a different color. So the sodium gates open. And basically this allows flow inside the cell or the axon. So just remember, if you forgot, I'm gonna draw this small picture here. I had this was the exterior, this was the interior, right? And then we had here, if you remember, I applied, and then I was measuring the outward current, and then here I had the sodium, right? And then the potassium was in the interior. So this allows basically sodium inside your axon. Okay, so this is the process until there. But then we have that from the process that I wrote above, there is some value in time at which the sodium gates now close, right? So you have this deactivation mechanism. So it's gonna decrease it. So while this is happening now, I'm gonna have sodium gates close and potassium opens. But when I say this, the potassium now, if the pump is allowing potassium to flow, it's gonna go from the interior to the exterior, right? So the flow is in the opposite direction. So you have to remember this. So I'm going to write here maybe a note. The potassium flow is in the opposite direction. Out of axon. Then what did we have in the, so here I told you that you're gonna have an overshoot, right? Past the V equilibrium for the potassium. So this is the V equilibrium, so it means that the potassium, while the potassium gates are open and they allow this exchange, we're gonna go a bit under the equilibrium, but then it's gonna tend to the equilibrium again, right? So it's gonna go under that for a bit, and then it's gonna come back up. So here we have the slow leakage. So is it gonna be in or out of the axon? The flow of potassiums. Exactly, it's the opposite direction, yes. Nice. Into the axon. And yeah, we have run out of time, but next time I will continue with these things. And it's very complicated, right? We have to keep track, we said, of N, M, H, and V. So it's a 4-dimensional thing. We don't want to work with very complicated things. So what we will do next is look at a different model that basically combines all this knowledge into a 2D model instead. So you can do phase plane analysis and whatnot. So it will be much easier to analyze. And yeah, thanks for today.